Welcome learners. Myself Dr. R. Suresh Kumar, Scientist C Molecular Biology Group, National Institute of Cancer Prevention and Research, Indian Council of Medical Research, Noida. Today we will see about epigenetics and its alterations. It is continuation with the IGNO lecture series for postgraduate diploma in environmental and occupational health. Before going to epigenetics, let's see the basics. So every cell in our body contains the same genome and same set of genes. Different organs have specialized cells and they have different functions. They have specialized functions. And we have in the cell, we have around 25,000 to 30,000 genes basically, which is there in the DNA. But these genes are not all get transcribed or it doesn't produce any protein. So every cell in your body expresses, and only small set of genes basically express certain genes. So during development, different cells express different sets of genes in precisely regulated fashion. So you have multiple organs and same DNA, but each organ has a specialized functions and accordingly genes are transcribed. So certain genes are on in particular organs and certain genes are off in the same organ, which is on in some other organ. So you see, this is what you have: all cells bears the same genomic DNA and same genes, and every organ has the same genes, same DNA, but only certain subset of genes are expressed in particular cells. So this is the animal cell structure, and you see that you have plasma membrane and inside you have nucleus, and inside nucleus you have DNA. And so you see this in the DNA basically organized into chromosome structure. It's a highly ordered structure. The DNA is packed with histone molecules, histone proteins, and they organized kind of bead on the string as kind of structures, and it form as kind of finger loops into, it is a multiple organization of different size and highly packed into a chromosome structure. So coming to the central dogma is that the DNA is a platform where the genetic material lies. It is a genetic material, and DNA is transcribed into messenger RNA. Basically, same language is scripted in a different format. Like if you see, English is taken a newspaper, same news is being given in different newspaper differently, but the same news. So the DNA is scripted into RNA, that is a transcription. This RNA is processed into a mature RNA, and this you have mRNA, tRNA and rRNA, and particularly this mRNA, they get into attached with the ribosome and they make a protein synthesis. So the message for the protein synthesis lies in the mRNA, and this mRNA message lies in the DNA. So as I said that all the genes, which is 30,000 genes we have seen, all the genes are not transcribed, all the genes are not going to produce a protein. Only certain genes are produced. So what is the mechanism? So at every step you have a regulation control. So different levels of regulation happens there in the eukaryotic gene expression. When you have DNA to RNA, there is a transcriptional control. From RNA transcript to final process, again processing control. From nucleus, when it goes to cytoplasm, that is transport control. Then mRNA that binds with the protein, that again translational control. So at every stage you have control process which actually controls a gene expression. So this is structure of gene. If you see the structure of genes, you have a I'm talking about from the left to right. You have enhancer, promoter, then actually a coding gene, that is called genes, what you call exons and introns. So genes are basically in the DNA exists as an exons and introns. The introns are intervening sequences, and the exons are the one which has a message for the protein synthesis. So there is the arrow which is called transcription start site. So the control of this production of this mRNA that lies in the upstream region, 5' upstream region of this gene. If you coming to the 5' region, that is a promoter and enhancer. The promoter region is the region where the RNA polymerase comes and binds, and enhancer is the one which actually enhances your transcription or decreases the transcription. The promoter region, where you see here, there is a lot of places, lot of areas in the promoter region which is under regulation. Sometimes this RNA polymerase comes and sits, and sometimes the RNA polymerase cannot sit. So this also makes the transcriptionally active, transcriptionally silent region. So if you see that this is a baby, nobody likes a baby to have a mustaches all over the body. So baby has to have a baby, but the baby has the same gene as adult has. So all the babies have the same gene. Why it is not having mustache? Basically, a gene which is meant for mustaches are suppressed at the infant age. When it comes to the adult age, this secondary sexual character appears. So that means the secondary sexual characters are silenced. The silencing is how? That's where epigenetic comes. So these are the different example, though this is not example of exact epigenetics, but it's for understanding purpose. A healthy baby has to express genes for infant age, but however certain babies they prematurely age, that is called progeria disease, that is aging abnormality. So there is when there is a problem in gene regulation problems, then this kind of disease state appears. So progeria is a premature aging. This is 7 years old boy which appears as a 69-year-old appearance. This is called progeria. So there is a spatial and temporal gene expression, what we call, and this one temporal is the time. So particular time only certain genes has to be expressed. Space is only particular space, one particular area the gene has to be expressed. I have seen that healthy baby and progeria, that is a temporal gene expression abnormality. Here coming to the next example, the spatial, this is a normal fly which has an antenna complex in the head, but people have mutated this, and during novel development they have changed the gene expression pattern, and they found that the leg appendages that formed in the antenna complex. So this is again mutation. This is a spatial abnormality, expression of genes abnormality. So there is a temporal and spatial gene expression is there. If this disturbed, then you have disease. So these all are set up not only genetics, but this all controlled by epigenetics. Let's see what is epigenetics again. So this is again cancer. So cancer also the same thing. Like in cancer, the people have abnormal gene expressions. You know, you have to two copies has to be there, but you'll have multiple copies, or the same copies are have transcribed multiple times. So this is abnormality gene expression also a problem in the cancers. You have tumor suppressor genes and oncogenes. The tumor suppressor genes basically present in the cells, in the normal cells, whereas absent in cancer cells. And oncogenes are absent in normal cells, which is present in the cancer cells. So this shut on and off mechanism that happens in the cancer cells due to epigenetics. This is what tumor suppressor genes, basically it has more tumor suppressor genes in the normal cells and oncogenes are less, so you'll have a normal cellular behavior. In cancer cells, when the tumor suppressor genes become less and more oncogenes, then cells abnormally behaves. So now there are so this tumor suppressor gene acts as a brake, you know, like in the car you have brake. So tumor suppressor genes they applies the brake for the speed, but oncogenes is pulls up, okay, go for more speed. You don't have to worry about this red light signals. So it is uncontrolled cell division that happens in the cancers. This all because of not only genetics, it also epigenetics. So I taken as an example this cancer as an example because we work on that. So the normal cells, they go for multiple stage carcinogenesis process. They initiated the carcinogenesis is initiated, and it become promoted, and it multiplies to progression, and it is metastasis. It is go to the multiple sites. So this multistage carcinogenesis takes around 15 years of this thing. 15 years of time for development, but during this development, all epigenetic factors, they play a role in alteration of gene regulation. So coming to the epigenetics. So now what is epigenetics? The term epigenetics was coined by C.H. Waddington in 1939 to name the causal interaction between genes and their products which bring the phenotype into being. Basically, he wants to say that the environmental factors, they interact with the genes and their products. So basically, environment gene interaction that results in the phenotype. If you see that nowadays people are giving a hormone injection to the chickens to have big mass. So basically, environment in the sense that hormone is environment now, either intercellular or intracellular or outside environmental factor that can influence the gene expression. So if you inject a hormone, a particular genes get expressed for muscle cells. So people inject chickens for premature muscle mass development. So this is again environmental influence. So it was later defined as heritable changes in the pattern of gene expression that are mediated by mechanism other than alteration in the primary nucleotide sequence of gene. In simplified, epigenetics is a study of heritable changes in gene expression that occur without altering change in the order of DNA sequence. So the DNA sequence, there is no any alteration in the DNA sequence, but the DNA sequence is modified. One of the nucleotides are modified, either addition of methyl group, or this modification is being altered into different chromatin organizations. Let's see what is the less level. So different types of epigenetic mechanism. One is DNA methylation, histone modification, and non-coding RNAs. These three different mechanisms that play a role in regulation of gene expression. We'll see that one by one what is the DNA methylation, histone modifications. DNA methylations are basically DNA methylations, addition of methyl group in the cytosine, basically that that happens in the DNA. And histone modification is that histones are the DNA is organized into histones. Upon histone modification, they become euchromatin, become heterochromatin, tightly packed chromosomes. So there is a non-coding RNAs are RNAs which also regulate the gene expression. So if you see that DNA methylation pattern during the development itself, single cell becomes zygote at 2 cell stage, 4 cell stage, 8 cell stage. At this cell stage, the 3 cell stage, from 8 cell stage, the 2 cell stage, 4 cell stage, these are totipotent. If you take a single cell, it can form a total two new organisms. But when it surpasses the 4 cell stage, it comes to 8 cell stage, it loses its totipotency. Now each cell destined to form a particular organ, in the sense that each cell are programmed to form a particular organ. So here in each cell a different epigenetic mechanism basically plays a role, and there is a DNMT, there are enzymes called DNMT3A, 3B and DNMT1 that actually forms a particular epigenetic signatures in the DNA that actually destined to form a particular organ. So basically epigenetics is this. So this set of epigenetic patterns should not be altered, they should not be disturbed. If you alter the set of pattern, then you have disease or any kind of abnormality in development. So 5 nucleotides that makes the DNA: adenine, guanine, thymine, cytosine. And if you say that cytosine is modified into 5-methylcytosine, methyl group is added in this. So this is a modification. This is what happens, modification in the DNA. And if you see that this is a normal gene, as I said that you have a gene exon, intron, and you have a transcription start site, and before transcription start site you have a promoter region. In promoter region you have a CGs, what you call cytosine followed by guanine regions are there. If the CGs are rich, that is called CPG rich regions. These CPG regions are amenable for methylation modifications. If these CGs are methylated, then transcription factors, what I have written as a TF, the transcription factors cannot come and sit if it is methylated. If transcription factor cannot come and sit, then the transcription won't happen. So the methylation basically silences gene expression, right? So the promoter region where it forms methyl group, this methyl group suppresses the RNA polymerase coming and sitting here by inhibiting some suppressive complex proteins. So coming to how this CpG island has a transcriptional control, as I see in the previous slide, it is basically the CpG rich regions in the promoter region, as well the CG regions which extends up to a first exon region that have a control over the transcription regulations. So these CG regions, if it is methylated, they won't allow the RNA polymerase come and sit, so that you won't have a transcription. So when you don't have transcription, particular protein is not produced. So this way some of the tumor suppressor genes are suppressed in the cancers, and that's how the DNA silencing happens. So this is an abnormality pattern of methylation pattern. So whatever we have seen that there is a methylation pattern which is set in the DNA that has been disturbed. So this is what in cancers new genes get methylated and new genes get demethylated. So this dynamics that changes that causes the cancer. As you see that this is a central dogma, we have seen the DNA and translation, and if you see the right side panel you have a different RNA, and you see that the normal in cancer you find a differential gene expression. You can see that some of the genes are missing by the arrow marks. So these are differentially expressed genes. So this is the cause, some of the genes are silenced in cancers by methylation process. So DNA methylation process, they have many roles. They have a genomic imprinting, exon activation. Basically this DNA methylation is needed for the normal physiology of the cells. many of the X inactivation, one of the X chromosome is inactivated by DNA methylation process, and tissue specific gene expression is by methylation process, sex determination and stem cell. So this is like pluripotent stem cells. When this CG rich regions in the pluripotent stem cells, when they get methylated, they become unipotent. So on the right side you have CG, the red color circles that shows that this is a methylated. Once if it is methylated, this is a particular cell type. And this is a particular cell type. If you disturb this methylation pattern, this cell type become a disease state. So in cancers they have deletion mutation, this is what they say. But in addition to deletion mutation of genes, methylation also one of the role for playing the carcinogenesis process. So we have many of the genes in the cancers identified which has a role in methylation. I mean which have been silenced by methylation, and DNA repair process, hormone response, vitamin response. These are the many genes which has been identified to be silenced by methylation process. And how this methylation has been happened? There are environmental chemicals, basically they induces methylation. There is a high arsenic contamination, cadmium contamination, nickel contamination, chromium. These are the contaminations. Those people who are exposed to this kind of contamination, they do get some of the genes get methylated and silenced. So because of this silencing of this due to this environmental pollutants, you do get diseases. These have been examples for environmentally induced methylation patterns. Now coming to the histone modulations, you know histones are the proteins which are H1, H2A, H3, H4. These are the 4 proteins. They are bound by DNA. This is a DNA actually wrapped around these histone proteins and form the chromatin structure. This binding is basically necessary for gene expression as well compact organization of the genes. So if you see that histone modulations, the histone is a protein basically. All the proteins get post-translational modulations. So post-translational modulation actually regulates the gene expression. Particularly H3 histone, they have some modulations like methylation, again protein methylation. This is a protein, not a DNA methylation. This is a protein methylation or acetylation of histone tails. They regulate gene expression. There are H3K4, the histone 3 and lysine 4 methylation, that is basically permissive transcription. But when this methylation become reduced, they become suppressive methylation. So like that there are some modulations, different modulations, they have different roles of methylation. I mean transcriptionally permissive, transcriptionally suppressive histone modulations. So this is what these are the compaction of histone molecules. When you have methylated histones or isolated histones, you find that euchromatin to heterochromatin structures in the down side of this template. So this is a euchromatin heterochromatin. How when you have histone modulations happen by acetylation or methylation, this interchange of euchromatin and heterochromatin happens. So there is another thing, acetylation. Acetylation removes the positive charge on the histones, results in repulsion. Basically DNA and protein, they bond by because of negative because of their opposite charges. But when this acetylation in the histones happens, it become negative. So DNA is also negative, histone also become negative, then they repel each together. So they won't bind tightly. So histone and DNA, when they are not binding tightly, that forms a transcriptionally permissible environment. So this acetylation basically induces transcription. Deacetylation basically silences transcription. So this is what happens when the acetylation is there. Then when there is the deacetylation, you have heterochromatin behavior in the histones. So this is what happens: euchromatin state to condensed chromatin state. So in the cancers, it can be euchromatin become heterochromatin or heterochromatin become euchromatin. So this is what happens, but this is coordinated function of DNA methylation and histone modulations. So coming to the non-coding RNA. Non-coding RNAs are RNA which doesn't code for anything. They doesn't code for any protein or something. They just exist as an RNA. Previously it was thought as a kind of RNA which is not having any function, but now it has been found that there are a lot of non-coding RNAs which have more than a protein coding RNAs. There are housekeeping RNAs and you have different RNAs, functional RNAs. Housekeeping RNAs are tRNA, rRNAs. There are the rRNAs which are necessary for the normal function. But there are certain non-coding RNAs which are having different sizes. The small RNA and long non-coding RNAs, they have different functions according to the cell type. So this is long non-coding, they function as a transport, they suppresses the protein expression, they take some protein to particular target, or they take other RNA to particular target. So they act as a different, these are the different function of this non-coding RNAs in the gene regulations. So mechanism of gene silencing, as we say that methylated and unmethylated. So the first graph that shows that methylated, they won't, they suppresses the transcription by methylating the cytosines, and also how it does is that they in the second you can see that they invite some of the repressive complex, which is called, we call by MECP1 or MECP2 complex. It is a complex of 5 proteins. They sit and bind with the methylated promoters, and they inhibit RNA polymerase to come and sit, and they stop the transcription. So these are the different the mechanism of gene silencing. And to methods to study epigenetics, for DNA methylation non-array based, and there is a candidate based method by PCR based, that is called methylation specific PCR, and methylation specific PCR combined with bisulfate restriction, that is COBRA we call, and restriction digestion based method. And the big way, if you want to do it, there is a methylation specific arbitrary primed PCR, representative different analysis, and MODITOF are the methods to do a PCR at this thing. And array based, nowadays most of things are array based and sequencing based. There are array based methods, are there methylation dependent immunoprecipitation methods, microarray, or NGS RNA, they are now methylation sequencing is there. So bisulfate sequencing, there are many methods are there to find out, and chromatin modulations can be done by immunoprecipitation methods. So this is a this is a methylation specific PCR. I'll just tell about 2-3 methods how you study the DNA methylation. If you want to study whether particular gene is methylated or not, so what will you do that? So first we do that, we take a DNA and we do sodium bisulfate treatment. Upon sodium bisulfate treatment, the methylated cytosines will be methylated cytosines, where unmethylated cytosines react with sodium bisulfate, and cytosines become uracil. You can see here in this diagram, this unmethylated DNA, you have ATCG, GTCA, TCG something, same sequence in methylated DNA. This methylated DNA is, this particular C is methylated, right? Whereas unmethylated is no methylation. This unmethylated DNA react with the sodium bisulfate. Then upon sodium bisulfate, they transform into uracil. Basically, it is a reaction, in vitro reactions we perform in the laboratory. Sodium bisulfate treatment, we do a sodium bisulfate treatment and some of alkali treatment also. Then upon final stage, we found the unmethylated cytosine become uracil. So we design a primer for methylated specific primer and unmethylated specific primer. Then we do the PCR. So coming to this methylation specific PCR, as I said that before, that after bisulfate treatment, the methylated cytosines become, it won't change into anywhere, but unmethylated cytosine become uracil. So now we set a PCR. So according to this changed nucleotide, we design a primer. So you can see this picture here, unmethylated cytosine and methylated cytosine, we have different primers and different temperature profiles for PCR. And down the line, if you see that the agar gel pattern in which that particular sample, say sample number 1, you have unmethylated and methylated. In unmethylated only you get a band, whereas in methylated you didn't get a band. So this means that both the alleles are found to be unmethylated. So you don't have methylation in this particular sample. So you can say that any patient sample you can receive and you can analyze particular gene methylation pattern. So same in the second sample, you see that unmethylated also you get a band, and methylation also you have a band. That means that one allele is unmethylated and another allele is methylated. And if you come to sample 5, you find only methylated band. In that sense, both the alleles are methylated in this case. So both the genes are silenced by methylation pattern. So this way you can find out what is the role of methylation in this. So why we have to study methylation is that if you have a methylation is a reversible modification. basically in the disease state, or if something you can reverse by some drugs are available which you can reverse this methylation for the time being. It can reverse this, like tumor suppressed genes, you can reactivate the tumor suppressed genes for the time being, and you can find out a cure or something. People are developing this methylation inhibitors, and there are methylation reactivation medicines are being under investigation for the translational part of this epigenetics. So next is how you study the histone modifications. How I will study histone modifications. We see that previously how DNA methylation has been studied by methylation specific PCR. Now we do histone modification. Histone modification basically how you study by chromatin immunoprecipitation technique. It is called chip assay we call. Chip assay basically, if you see that the first box I have DNA is there, you take a genomic DNA, and basically when you isolate DNA, you have to isolate DNA in such a way that your DNA and protein interactions won't get harmed. So when you isolate your cells, we actually we fix the cells in formaldehyde so that the DNA protein interaction won't get disturbed. So once the fixation has been done, then we isolate DNA, then wherever the protein is bound, it has been intact. So now we put a restriction enzyme or physical shearing to fragment the DNA, the second box. When you fragment the DNA, you have multiple fragments of DNA being there, and the protein of interest, say like your protein of interest. So I have antibody against this particular protein, and I'll put an antibody against this particular protein, and wherever this protein is bound, so only those regions are bound by antibody, and I immunoprecipitate those. This particular antibody is magnetic labeled so that I can pull down with a magnetic precipitation. Once it is pulled down, I reverse cross link so that the protein is being removed. So whatever the DNA element which has been bound by this protein can be sequenced, and you can find out which regions are bound by this protein. Now, as I say that histone molecules are post-translational modification, they have post-translational modifications like H3K4 trimethylation. So I'll have a antibody against H3K4 trimethylation. I have antibody for H3K27 methylation. I have acetylation, and I have a antibody against particular area. So that way you can put these antibodies and you can immunoprecipitate and look for the regions which has been bound by this kind of modulations. So in this way you can find out a genomic landscape, what are the proteins what are the genes have been regulated by this histone modulations. So again you have histone modulations, you have a lot of medicines have come up now, you can reverse this histone modulations. So once you get this basic idea of that what is the role of this histone modulations, you can really look for some of the advantages for therapeutic purpose or any methods. So these kind of things are always possible in any kind of applications in other also. So non-coding RNA, basically I say that non-coding RNA basically you can do by real-time PCR based method, like again it's a PCR based method which I am not going to tell here. So you can have a PCR based method or RNA sequencing based methods which can isolate RNA and do a RNA sequencing, and you can find out what are the RNAs have been differentially expressed. So one can find out what are the long non-coding RNA differentially expressed in particular disease state, and you can see that what is the role of that. So summarizing all this is basically epigenetic traits are heritable phenotypes resulting from changes in chromosome that do not involve changes in DNA sequence. So the scientific and medical interest in epigenetics has increased dramatically in recent years due to pathogenesis caused by environmental factors, lifestyle changes like tobacco smoke. Chronic tobacco smoke, people have different epigenetic changes. People who are consuming pollutant water, they have different diseases due to, and that is induced by epigenetic factors. So three prominent epigenetic mechanisms that involves DNA methylation and histone modulations and altered non-coding RNA expressions. DNA methylation, methylation at cytosine of promoter region causes gene silencing, and histone modifications, acetylation relaxes chromatin, and methylation also has a role in histone modifications, and deacetylation suppresses gene expression. Histone H3, K4, K9, K27 are hotspots for modification that are regulated by specific modulators.